Good afternoon, ladies and gentlemen. My name is William Wei-Tse Hsu. Today, I'm going to introduce you our research about exploring the inhibitory activity of brilliant blue G toward the formation of amyloid fibrils derived from lysozyme. In this presentation, I will first give you some backgrounds related to this project. Then, moving to the motivation and objective, I will focus on potential therapeutic strategy for protein aggregation diseases. Also, the methods and the results of this research will be included. At last, I will present a summary of this research. Protein aggregation is a general property of all proteins and can occur under appropriate conditions. In the field of biomedical engineering, molecular biology, neuroscience, and protein engineering, protein aggregates, which are also called amyloid fibrils, have been proved to be associated with some human diseases, which are also called amyloidosis. As you can see, different kinds of protein aggregates can cause different kinds of human diseases. So, what is the relation between amyloid fibrils and amyloidosis? In this flowchart, you can see that unaggregated protein can become folded and become amyloid fibrils. These amyloid fibrils are similarly in shape of fibers and are abundant in beta-sheet structure. When these amyloid fibrils deposit near neurons or cells, they may therefore cause cell death and lead to fatal diseases. Therefore, to decrease the occurrence of amyloidosis, scientists have devoted themselves to developing possible treatment against them. Fortunately, previous researches have shown that using small molecules is one of the potential therapeutic strategies for treating protein aggregation diseases. The inhibiting ability of this small molecule is employed to mitigate the fibril formation. Our research, therefore, is to investigate another small molecule, brilliant blue G, and to examine its inhibiting ability. As for the objectives of our research, we take lysozyme as our model system and BBG as our chosen small molecule. First, we want to understand the effect of the BBG on lysozyme fibril formation. Then, we speculate the inhibition mechanism of amyloid fibril formation. In the end, we hope that our research can be helpful to develop cheaper alternatives to tackle protein aggregation diseases. As for the approaches that we used in this research, first, we used several spectroscopy to examine the formation of amyloid fibrils. Then, we used TEM, circular dichroism spectroscopy, to verify our observation. In the end, we used molecular docking and molecular dynamic simulation to predict the potential binding sites and the interactions between BBG and lysozyme. In this part, I will divide our results into 6 parts with 6 questions. So, when examining the effectiveness of BBG, the first question is whether BBG is capable of inhibiting amyloid fibril formation. To answer this question, we have to turn to the plot at the left. We incubated lysozyme with different concentrations of BBG under 55 degrees Celsius and pH 2 condition. Then, we conducted THT fluorescence assay at different incubation times. To analyze the results of this experiment, first, we have to know the fact that because the THT can bind to the amyloid fibrils of lysozyme, the intensity of THT fluorescence is therefore proportional to the amount of amyloid fibrils. As you can see in the figure, when the concentration of BBG increases, the intensity of THT fluorescence decreases. Therefore, we can know that the amyloid fibril formation is inhibited effectively by BBG. Moreover, we took some TEM photos of lysozyme. As you can see in the figure, you can see that the formation of amyloid fibrils of samples with BBG is obviously less than those without BBG. Therefore, from the results of THT fluorescence assay and TEM photos, we can know that the amyloid fibril formation is inhibited by BBG effectively. To take a closer look at the inhibition mechanism, we want to know that whether BBG can affect the secondary structure of lysozyme. To answer this question, we conducted a circular dichroism spectroscopy. As mentioned, amyloid fibrils are abundant in beta-sheet structure. That is, there is a positive correlation between amyloid fibril formation and beta-sheet structure. As you can see, when the concentrations of BBG increases, the proportion of beta-sheet structures decreases. Therefore, we can know that amyloid fibril formation is suppressed by BBG. After knowing that BBG can affect the secondary structure of lysozyme, in the third question, we want to know that whether BBG can affect the tertiary structure and hydrophobic areas of lysozyme. To answer this question, we conducted ANS fluorescence assay. ANS is a molecule that can bind to the hydrophobic surfaces of proteins and is therefore used to detect the partially folded states of proteins. When proteins start to aggregate abnormally, they lose their native states and the hydrophobic surfaces are exposed, causing the intensity of ANS fluorescence to increase. As you can see, at the beginning, the intensities of all samples are nearly zero. However, at day 7, you can see that samples without BBG has the highest intensity of ANS fluorescence. On the other hand, the samples with different concentrations of BBG have lower intensities of ANS fluorescence. Therefore, we can know that with more BBG added, less hydrophobic areas will be exposed, and the amyloid fibril formation is therefore suppressed by BBG. As far as I have mentioned in this presentation, we have consolidated the inhibiting ability of BBG on amyloid fibrils formation from an experimental perspective. However, if we want to have a better understanding of inhibition mechanism, we have to analyze our subject from the molecular perspective. That is, we use simulation software such as Discovery Studio and GORMAX to conduct molecular docking and molecular dynamic simulations. So, by using these simulation softwares, we are able to answer the question that whether BBG can affect the stability of lysozyme. To answer this question, we have to observe two parameters. The first parameter is RMSD, root mean square deviation. RMSD averages the deviation of every atom when comparing with the initial position of the structure. As you can see, RMSD of lysozyme sample with BBG is much smaller than that of lysozyme without BBG, implying the greater stability of lysozyme with BBG added. Therefore, from the results of RMSD, we can know that a lysozyme is indeed stabilized by BBG. Compared with RMSD, RMSF, which is root mean square fluctuation, measures the averaged fluctuation of atoms in a given time. As you can see, RMSF of lysozyme sample with BBG is much smaller, implying a greater stability. In addition, it can be seen that the fluctuation of residue 115 to residue 129 is reduced significantly. This sequence, we therefore speculate that this region may be strongly related to the inhibition mechanism. And also, we can know that lysozyme is indeed stabilized by BBG from the result of decreasing RMSF. After knowing the variation of stability of lysozyme, in the fifth question, we want to know the interactions between lysozyme and BBG. To answer this question, we conducted a molecular docking using Discovery Studio. As you can see, different kinds of interactions can be predicted in molecular docking. Also, you can see that hydrophobic residues such as alanine 10, leucine 129, and valine 120 are exposed because of 55 degrees Celsius and pH 2 condition. So our last question is, what is the possible inhibition mechanism of BBG against the formation of amyloid fibrils? From the results we obtained from the molecular docking, we can see that there are obvious interactions with region 115 to 129, which is shown to be important from the result of RMSF, as mentioned in the previous part in this presentation. Also, we can see that there are significant interactions with hydrophobic residues, which are exposed because of 55 degrees Celsius and pH 2 condition. Therefore, we speculate that BBG inhibit the fibril formation by interacting with region 115 to 129 and interacting with hydrophobic residues to prevent further exposure. In conclusion, with addition of BBG, we can know that the THT fluorescence intensity and ANS fluorescence intensities and the proportion of beta sheet structures are all decreasing. Also, we know that the BBG can stabilize the lysozyme from the results of RMSF and RMSD. Also, we know that the region 115 to 129 and hydrophobic residues are affected by BBG. Based on these results we obtained from the experiment and simulation, we can therefore draw three main conclusions. First, BBG indeed inhibits the formation of amyloid fibrils. Second, BBG inhibits amyloid fibril formation by influencing sequence 115 to 129. Third, interacting with hydrophobic residues, BBG prevents further unfolding and aggregation, therefore prevents the formation of amyloid fibrils. This is the end of my presentation. Thanks for your listening.